Good afternoon, everyone. It's the top of the hour, so we wanted to get started with today's webinar. So we'd like to welcome everyone to today's mini symposium. My name is Marie Latalipe, and I manage some of the nutrition programs at our organization. And this series is brought to you again in collaboration with the American Society for Nutrition, and it's organized specifically by our sodium committee. So for anyone not familiar with us, we are a nonprofit scientific organization. We advance nutrition and food safety science for the benefit of the health of the public. And we always like to emphasize that we do not lobby. And the way we operate is that we bring together the intellectual contributions of scientists from industry, academia, and government. And then together they advance science in areas that are of common interest. So we really want to thank everyone for joining today. We have a really interesting couple of speakers. Just to note that this will be recorded and it will be available on our website if you'd like to go back to that as a resource. And as we go through the presentation, we will likely take the questions at the end, but please feel free to type any of your questions as we go through the two presentations. So with that, I am very pleased to introduce our first speaker, Dr. Shannon Lennon. Dr. Lennon is an associate professor at the University of Delaware in the Department of Kinesiology and Applied Physiology. Dr. Lennon's lab, which is the Cardiovascular Nutrition Lab, specifically studies the role of nutrition on cardiovascular health and disease, and her group routinely conducts controlled feeding studies and is actually currently funded to study the role of dietary potassium in attenuating the effects of a high sodium diet on the vasculature. And she's been a registered dietitian for over 20 years. So with that, I will turn the platform over to you, Dr. Lennon.
Thank you, Marie, for that kind introduction, and welcome everyone. I apologize that my webcam is not working with the particular software we're using, so my apologies, but hopefully everyone can hear me clearly. All right. So I'd like to start with just a little bit of an introduction on the human microbiota. I'm not sure everyone's knowledge in this area, but as humans, we contain roughly 100 trillion microorganisms that belong to the various domains shown here on the left side of your screen. And they populate everything from our oral cavity down to our urinary tract. However, our intestines, small and large intestine, are most heavily populated. Now, there's roughly 10 main phyla that are found within the human gut microbiota. However, we are most populated by these first four. So we're looking at Firmicutes, Bacteroides, Actinobacteria, Proteobacteria, and sometimes people do include Microbia. So in most cases, a commensal relationship does sort of exist between our gut flora and us as humans, and in some cases, this is actually mutualistic. For example, gut microorganisms can help ferment our dietary fiber that we consume into short-chain fatty acids. It can also help us meet part of our requirements for some of the B vitamins, as well as vitamin K, and also plays a role in metabolizing bile acids, sterols, and xenobiotics. It's important to note that the composition does change over time as our diet changes, as well as our overall health. And when there is an imbalance in the gut microbiota, we often refer to this as gut dysbiosis. So there's some common terminology that I want to review with you before I dive into some of the literature, just to kind of set the stage. So we often see alpha diversity represented in many of the papers that we'll look at, and this is looking at diversity within a sample. For instance, looking at the richness in terms of the number of different microorganisms that may be present in a sample, as well as the evenness. So how even are they relative to one another? And the Shannon Diversity Index is one that's often used. This actually combines both richness and evenness. The chow richness is also reported often. It's generally an estimate of the total operational taxonomic units that are present in a given community. And those OTUs are often used to categorize the bacteria, really looking at the similarity of their sequences. And finally, the PLAU evenness looks at the evenness within a community. So if you look at the circles to the right of your screen, we can see if you look at this top circle here, we have 2 blue circles and 2 green stars. So we're looking at 2 different species within this sample, giving it an alpha of 2. Now, beta diversity would look at diversity between samples. So for instance, between the top circle and this lower left-hand circle. Some of the metrics that are used to represent beta diversity include the Jacquard index, which can look at the presence or absence of distance between samples. The Bray-Curtis will actually weight the abundance and look at that distance between samples. And oftentimes this is represented graphically using a principal coordinate analysis. So if we jump back over to our circles here on the right side of the screen, again, if we look at the top circle, we see that we have sort of two different species here. If we look at the lower right hand, we see we have two red circles, we have a blue square and a yellow star. And if we look across these two samples, we see 5 different species represented, so a beta of 5, if you will. So let's dive into the literature and see where sort of some of this information in terms of gut dysbiosis and hypertension first appeared. So there was a paper published back in 2015, so this is obviously not that old, that really asked the question, does gut dysbiosis exist in a hypertensive rodent model? So in this case, they used the spontaneously hypertensive rodent, or SHR, and they compared it to a normotensive control, the Wistar coyote. And they had about 5 and 6 animals in each group. The first thing they did is they looked at the fecal biomass in terms of the DNA, and they saw that the bacterial load within the fecal samples was greatly reduced within the hypertensive rodent compared to the Wistar coyote control. And the hypertensive here is in red, and our normotensive Wistar coyote is in green. They also saw a significant difference in the P-Low evenness, the chow richness, and the Shannon diversity indices, all indices of alpha diversity, so diversity within a sample. And so this was reduced in the SHR compared to the Wistar coyote. This has been shown in other models as well, so the deoxycorticosterone acetate / salt rodent, as well as the stroke-prone SHR rodent and the Dahl salt-sensitive. So this has been replicated in other hypertensive rodent models. On the right side of your screen, we're seeing a PCOA here, a principal coordinate analysis, and you can see that the samples from the Wistar coyote cluster closely together, and the SHR samples cluster closely together, and they're kind of separated apart, showing differences in the diversity between these two groups. Now, within this study, they also did a small pilot project, if you will, within humans. They recruited 17 adults, and they categorized them as either being hypertensive or not. And they used a blood pressure of 125 millimeters of mercury or higher for a systolic to kind of define that. And so when we look here, we can clearly see that the systolic blood pressure is greatly elevated in those individuals that were labeled hypertensive, and significantly reduced in those that were considered control. We don't see any difference here in the P-Low evenness. However, there is a reduction in the chow richness and Shannon diversity indices, again, indices of alpha diversity. And so, again, this highlights not only in rodents, but also in hypertensive adults, that microbial richness and diversity is reduced. They also saw this with an increase in the Firmicutes and Bacteroides ratio. And these two are two of the major phylum that we find within the human gut microbiota. And in this case, we're seeing an increase in the Firmicutes and a decrease in the Bacteroides, hence shifting that ratio. Now, when they plotted these samples to look at beta diversity, again, they saw that the control subjects were clustering together and the hypertensive were clustering together. However, we see 2 green circles appearing here among the hypertensives. One of the things that was noted in this paper is that they were categorized based on their current systolic blood pressure. So subjects could have had a history of hypertension, but if their blood pressure, their systolic was below 125, they were labeled as control. So I suspect that was the case with these 2. Now, further work that really sort of linked the gut potentially to hypertension is a study that was done in germ-free mice. So germ-free mice have a sterile gut, so there are no microorganisms populating their GI tract, and they were compared to conventionally raised mice, so those that would have, microorganisms in their gut. And both groups were challenged with angiotensin II. So mini pumps were attached to these animals, and they were infused with angiotensin II for 7 days. Angiotensin II is a potent vasoconstrictor and can lead to the development of hypertension. So in these animals, they actually removed the aorta of these animals, and they looked at how the aorta actually responded to these various vasodilators. So what you're looking at here is a curve that plots increasing doses of acetylcholine, which is the vasodilator, and we're looking at percent relaxation of the aortic, or in this case, aortic ring, in these 4 different groups. The black lines represent the conventionally raised animals, and the red represent the germ-free mice. And what you see is that this particular group of conventionally raised mice that was treated with ANG2 doesn't relax quite as well, suggesting that they have vascular dysfunction. When they also looked at systolic blood pressure in these 2 groups, conventionally raised mice in black, germ-free in white, they can see that in the presence of ANG2, we have an increase in blood pressure in these conventionally raised mice, but we do not in the germ-free mice here. And so what this suggested was that the gut microbiota was needed for the hypertensive response to angiotensin II. And finally, there was an additional experiment done in germ-free mice who received fecal material transfers from hypertensive adults. So what they did is they recruited a control human, had a normal blood pressure. They had 2 humans that had hypertension, so they had an elevated blood pressure here, and they collected fecal samples. And then they actually transplanted those into these germ-free mice. After 7 days, they collected fecal samples from these mice, and 9 weeks later, they looked at their blood pressure. And what they saw is in those mice that received sort of the fecal material transfer or transplantation from these hypertensive adults had an increase in systolic and diastolic blood pressure compared to the germ-free mice that received this from a control or normotensive human. They also looked at parameters of alpha diversity. So here we see the Shannon index. This is greatly reduced in those germ-free mice that got the FMT for the hypertensive adults. And we also see when we look at this sort of on a PCOA that there's a close clustering of these samples in those germ-free mice getting the hypertensive FMTs versus those that got the control. And so what this suggested was that the gut microbiota can be transmitted and it can elicit a hypertensive response. One of the things that I do want to note is while this paper was published in 2017, these findings actually haven't been validated or replicated by another group. So while they're very interesting, hopefully we can see another group come out and kind of validate these findings here. So just want to highlight that through some of the cross-sectional work that's been done in hypertensive and normotensive adults, there are some common genera that appear to be altered in hypertensive adults, and they're listed here. And given the challenges with some of these names, I'm not going to go through and pronounce all of them, but there have been some common genuses that have been highlighted as being altered in hypertensive adults, as well as some lower levels of genera, meaning that they're not quite as abundant as others. I do want to highlight that many of these are short-chain fatty acid producing bacteria, which will be part of the focus of our second talk by Dr. Zhu. So I don't want to steal any of her thunder, and I'll let her, explore that area a little bit more. So this talk is not just about gut dysbiosis and hypertension, but it also involves sodium. And so why do we include sodium? Well, I don't think it's any secret that sodium consumption is linked to an elevation in blood pressure. The graph to the right here shows the typical average sodium intake in the American diet, roughly 3,600 milligrams per day. So this is a combined value between men and women. It greatly supersedes the dietary guidelines for Americans that recommends no more than 2,300 milligrams per day, and certainly supersedes the American Heart Association's guidelines of 1,500 milligrams per day. In addition, increased sodium consumption in the diet is linked to an increased risk for stroke and heart failure, as well as other non-cardiovascular related conditions such as osteoporosis and kidney disease. One thing that I really want to highlight here is the gut is the first as well as the largest location for sodium absorption. So what we eat in our diet, our gut is really the first part of our body that is exposed to that. So naturally, the question becomes, could a high sodium intake modulate our gut microbiota composition? So there was a paper published in Nature in 2017 from Dominic Mueller's group out of Germany, in which they gave mice a high sodium diet. So they had two groups. They had one group that received a normal sodium diet. This is shown by the white bars, and another group that received a high sodium diet. This was a 4% sodium chloride rodent diet here for 14 days. And they looked at the relative abundance of a number of different genuses here. And what they noted was that lactobacillus was significantly reduced, pseudoflavor factor, as well as Clostridia, while Parasudorella was elevated. Now, they were particularly interested in the lactobacillus genus, and they started to look at some of the species within this genus. And here we're looking at the relative abundance of Lactobacillus marinus. What we can see here is we can see nice abundance of this particular species on the normal salt diet, which then dramatically falls, and we see a reduced abundance during the 14 days of the high salt, which seems to start to rebound once the mice are returned to their normal salt diet. Now, additionally, they also looked at systolic blood pressure using telemetry in these mice. So you can see here systolic blood pressure on the normal salt diet, increases in the high salt diet. And then in the subgroup of these animals, they actually supplemented them with Lactobacillus marinus for 24 hours. And we start to see a drop in that blood pressure, which is illustrated a little bit more clearly here on the lower part of your screen. So we're looking at systolic blood pressure here and diastolic blood pressure here. Again, the white bars are the normal salt diet, dark gray is high salt, and then these lighter gray represent the supplementation with Lactobacillus marinus. So we start to see a decline in the systolic and diastolic blood pressure. Now, additionally, in this paper, they also did a small pilot study in humans. They recruited 12 men between the ages of 18 and 50, and they put them on a high salt diet. And what they actually did was they supplemented their diet with salt tablets, which brought their sodium intake up to 6,000 milligrams per day. What they did note in a number of their subjects is that they started to experience nocturnal hypertension. So during the nighttime hours, they started to see an increase in both systolic and diastolic blood pressure. Now, given their work in the mice in which they were focused on the Lactobacillus genus, they looked at a couple of species within this genus across 5 of their subjects. And so just to orient you to this graph here, the black rectangles here represent the values on the baseline diet. The gray rectangles here represent on the high salt. If there happens to be an X in that box, that means there's a complete loss of that species. They also highlight species abundance using a heat map, so the darker the color, the greater the abundance. And what we can see is that for a number of these species, we see a complete loss of the various species with the high salt challenge here. So again, Lactobacillus as a genus seems to be vulnerable to the high salt challenge, particularly when we look at various species here. So also involved in this story is the immune system. And there was a recent review article that came out this year that really hypothesized how the gut microbiota, as well as inflammation, might play a role on the development of salt-sensitive hypertension. And so what they highlighted is that this excess salt intake seems to activate a number of pro-inflammatory macrophages. In particular, here we're looking at a dendritic cell within the gut. We see an increase in reactive oxygen species, leading to an increase in T cell activation. Now, naive T cells can differentiate into a variety of different types of T cells, but in particular, we're interested in the T helper 17 cells and the T regulatory cells. Why is that? Well, Th17 cells have played an important role in protecting us from bacterial and fungal infections. However, they also have potent inflammatory potential, in particular, secreting a number of interleukins. And there has been evidence to suggest that salt can increase an induction of Th17 cells, in particular releasing this interleukin 17, which may then play a role in salt-sensitive hypertension. Simultaneously, T regulatory cells, which are important to sort of regulate and suppress the immune system, seem to become impaired under high salt conditions. So not only do we have the impact of the salt on sort of the gut microbiota, but there seems to be this interplay with the immune system, which is also perhaps contributing to the development of hypertension. So what's the evidence for this? Well, Dominic Mueller's group in their study in those 12 males collected peripheral blood mononuclear cells from about 8 males before and after the 14 days of that high sodium diet. And what they looked at is CD4 positive, so T cell positive, interleukin 17A positive, and TNF alpha positive cells. And they saw that this was greatly increased under high salt conditions, suggesting that sodium loading is inducing an inflammatory response. Now, this had been previously shown several years ago, back in 2013. Kleinvietfeld had isolated peripheral blood mononuclear cells from healthy volunteers. Those were cultured, and then they added sodium chloride to the culture, and they did see this increase in IL-17A. So this kind of was precursor work to what's seen by Dominic Mueller's group more recently. So most of the human work that I've shown you to date has been rather small, sort of either cross-sectional studies or small pilot studies. And really, you know, we don't have much out there, I will tell you, in this area. But there was a paper published earlier in the Journal of the American Heart Association from Finland that looked at a cohort of about 7,000 individuals. And so they were really interested in looking at whether those hypertensive adults within their cohort had an altered gut microbiota. So if we look at the panel up here, we're looking at a couple of parameters related to blood pressure, such as systolic blood pressure, diastolic, pulse pressure, mean arterial pressure, and hypertension. And this was looked at whether there were associations with alpha diversity. In this case, they were actually looking at the Shannon Diversity Index. And what they saw was that standard deviation increase in the microbiota alpha diversity was inversely associated with systolic blood pressure. They also saw some relations here for beta diversity, okay? And I should highlight here that in this first model, they adjusted just for age and sex. Now, when they added other covariates into this model, I believe they added body mass index, physical activity, smoking, medication. Many of these associations were lost, and I should highlight that most of the changes that were found in the age and sex adjusted model were predominantly in the Firmicutes phylum, which is one of the major phylum found in the human gut microbiota. Now, given the work by Dominic Mueller's group looking at lactobacillus and the various species within that, they were interested in exploring this further in their cohort. And they did have 24-hour urinary sodium excretions in many of their subjects, which is an excellent indicator of sodium intake in the diet. And so they kind of looked to see if there was any associations here. They did not see any with lactobacillus, the genus. However, they did see an association with lactobacillus paracasei here. So a greater abundance of lactobacillus paracasei was associated with a lower mean arterial pressure and a lower sodium intake, while lactobacillus salivarius had the opposite relationship. They also did look at men and women separately. They had over 3,000 men and women in this cohort, and they were looking at a number of species within lactobacillus, and again, whether there was an association with various blood pressure parameters. To the right here is a heat map. So the darker the red color, the greater the increase, the darker the blue color, the greater the decline in that species. And we can see there tends to be more associations showing in women than in men. Most of the colors are not very strong, so not real strong associations, but definitely, you know, something going on with many of the species in lactobacillus here in these hypertensive individuals. And then really I think the final paper in humans that I want to share with you that came out in 2019, again, this was a cross-sectional study in which they recruited about 135 healthy volunteers, and they categorized them into a low or high salt consumer group based on a 3-day food record. So those that consumed 2,300 milligrams of sodium per day or less were considered sort of the low sodium consumers, and those that were above this were considered the high sodium consumers. And you can see that most of their subjects did fall into this higher sodium consumer group. There were 93 out of the 135 as opposed to 39 in the other group. Now they were well matched for age, there were more men in the higher sodium group, which is not unexpected. Typically, when you consume a little bit more energy, your sodium intake also goes up, pretty evenly distributed across the races here. Now, when they looked at blood pressure, they did note that systolic and diastolic blood pressure was higher in the high sodium consumer group, although still within the normotensive range. They did tend to have a higher BMI, and obviously their sodium intake was greater. And so they used two different dietary recall methods. They used a 3-day food record, which was reflective of their short-term intake, and the subjects also filled out a food frequency questionnaire, which was reflective of their long-term intake, so kind of what they had consumed over the last 12 months. I want to highlight here panel C, which shows the Shannon Diversity Index here, again, looking at alpha diversity. They did not see any difference between these two groups. However, they did see a difference in the beta diversity shown here in panel D. So they looked at a number of different genuses, species within this particular study. And while in their table they talk about short-term and long-term intake, in the graphical representation of the different microorganisms, they use the term observed sodium intake, which is reflective of the 3-day record, and self-reported sodium intake, which is reflective of that food frequency questionnaire. And you can see that, you know, for some of these microorganisms, we do see only increases with the FFQ or the diet record. Some are consistent across both here. So there's a little bit of inconsistency, which does happen with utilizing different dietary records or recall methods. However, overall, they did see that a higher salt intake was associated with an increase in Firmicutes, proteobacteria. These are two major phylum, as well as the genus Prevotella, and they were all associated with an increase in blood pressure. And they did plot sodium intake relative to systolic and diastolic blood pressure for both the 3-day record and the food frequency questionnaire. And they clearly saw a difference in systolic and diastolic with the 3-day record. However, they only saw a difference in systolic blood pressure with the food frequency questionnaire. They did not see that with the diastolic blood pressure. So the last thing that I want to share with you is something that came out of Binna Jo's group at the University of Toledo. Her group has done a lot in this area, particularly using the dull salt-sensitive rodent as their model. And one of the things that they were interested in looking at is, you know, whether there's a matching between sort of the diurnal rhythm that seems to exist with the gut microbiota as well as blood pressure. So blood pressure does have a very well-defined rhythm. It tends to be highest during our awake hours, particularly in the morning, and it roughly drops about 10 to 20% during the night. And this is known as sort of blood pressure dipping. And actually, if you dip less than 20%, you're identified as a non-dipper, and it's thought to carry an increased risk for cardiovascular disease. And there's been some evidence to suggest that the gut microbiota may also have a similar diurnal rhythm, and they wanted to see if that was actually the case. Now, I just want to remind everyone that rodents are nocturnal animals. So during the nighttime hours, they're awake and active and consuming their diet, while during the day, they're resting asleep. So they're obviously opposite to us. So in this particular study, they utilized the dull salt-sensitive rodent. And with the dull salt-sensitive rodent, when you feed them a high salt diet, their blood pressure goes up. And so they had two groups here, one group that received a low salt diet and another group that received a 2% sodium chloride diet. And what you're looking at here is sort of a 2-day period in which they evaluated systolic blood pressure for those animals on the low salt diet and those animals on the high salt diet. The sort of gray bars here represent the nighttime hours, and the open white bars represent the daytime hours. And so you can see that their blood pressure goes up during the night when they're awake and active, and it falls during the day. You can see that those animals that received the high salt diet had a greater increase in their blood pressure, and then it falls a bit more. And so when they actually looked at their dipping, we see a greater dip in the high salt animals just because their blood pressure goes much higher during the day. Now, during this, they collected fecal samples once during the nighttime hours and once during the daytime hours. And so they were interested to see, was there an association with these changes in blood pressure? And so they looked at some of the major phylum. We see Firmicutes here, Bacteroides, and Actinobacteria here. And what they saw is that when you look at sort of the nighttime hours here, the low salt group is in the gray bars, the high salt is in the red bars. No difference here. When we go to the light hours, when they're resting, we can see an increase in both groups, but again, no difference between whether animals received a low salt or high salt diet. And this was consistent across the different major phylum that they looked at, which really suggested that it's the timing of day, but not really the salt that impacted the diurnal timing on the gut microbiota. So fortunately, while their hypothesis was not proved true, it was kind of interesting to see that there are some fluctuations in these major phylum from our awake hours versus our nighttime hours, at least as shown here in these rodents. So I just want to finish up and share a little bit about our current work. This is an area that we've been getting into over the last year and a half. I have a senior doctoral student, Katerina Smiljanets here, whose dissertation is focused on looking at the effects of high dietary sodium intake on gut microbiota composition in both men and women. And we were interested in doing this study because there really is very little interventional work looking at various sodium intake, as well as looking at both men and women. So we were interested to look at the gut microbiota composition when an individual is consuming a recommended sodium intake of about 2,300 milligrams and how that might be altered when their sodium intake is elevated. We were also interested in looking at their immune response, so focusing on the Th17 cells and T regulatory cells. And we were also interested in seeing whether there were any sex differences between men and women in their response, particularly in the gut microbiota. So this is our current study design here. CAT is recruiting 28 normotensive men and women, and so 14 men and 14 women. And so they'll initially either be randomized to a recommended sodium diet or a high sodium diet. So the way that we're running this is that all subjects will consume a recommended sodium diet. However, they'll either receive placebo tablets or salt tablets that would bring them up to about 7,000 milligrams per day. And they'll follow this diet for 10 days. On the 10th day, we're collecting blood to assess immune markers, as well as fecal samples to look at the various species there. Subjects will undergo a 14-day washout, and then they'll cross over to the other arm. So this is a randomized crossover design trial here. Fortunately, with the pandemic, we had this trial up and running early in the spring, but at the University of Delaware, we were shut down in mid-March, and we've really only started back in mid-September. And so CAT's made great progress. We have, I think, at least 20 subjects through now, so I'm hoping that we'll be sharing our results with you later in 2021. So just to summarize here, with high sodium diets, we see some gut dysbiosis showing up, and particularly individuals with hypertension. I think this has been clearly shown that gut dysbiosis exists in hypertensive rats and humans. Salt loading has been shown to lead to gut dysbiosis in mice, and we're currently trying to replicate that, if you will, within humans. With some changes in the gut microbiota, we particularly see changes within the Firmicutes phylum overall. We see increases in the Prevotella genus, and certainly the Lactobacillus various species are reduced. We also see some changes overall in richness and diversity here. But it's important to note that most of the work is cross-sectional or pilot studies that have been limited to predominantly men at this point. We also are interested in the relation between sodium and the immune system and how this may have implications for the gut microbiota. In particular, we're interested in these Th17 cells and T regulatory cells, and there's been evidence to suggest that they play a role in the development of hypertension. But I do want to highlight that really we need many more human interventional studies, including both men and women, in normotensive and hypertensives, that where really this is a very new area. As you see, many of these papers only date back to 2015. So there's a lot of work that really needs to still be done in this particular area. So with that, I just want to highlight my cardiovascular nutrition lab group at the University of Delaware. This is Katarina Smiljanets, who's leading our work in this area currently, and the various individuals that populate and where we call home here when it's not the pandemic and we're in work every day. And with that, I guess we're doing questions at the end, but I thank you for your attention.
Great, thank you so much, Dr. Lennon. I think, as you mentioned, we'll go right into our next speaker and take the questions at the end. So I am very pleased to introduce Dr. Haidong Zhu. Dr. Zhu is a molecular geneticist and a professor at the Georgia Prevention Institute at Augusta University. And her research programs over the past 20 years have focused primarily on two things: the interplay of genetics, lifestyle, and nutrition in salt-sensitive hypertension and cardiovascular disease, and then also the effects of lifestyle and nutrition on cell aging, epigenetic aging, and the prevention of aging effects. She has over 95 peer-reviewed publications, and her research has been supported by the National Institutes of Health, as well as the American Heart Association. So today, Dr. Zhu is going to present her recent work, Dietary Sodium, Short-Chain Fatty Acids, and Blood Pressure Control in Untreated Hypertensives. And this paper was actually selected by the journal Hypertension as the high impact paper for fall 2020 in the category of clinical science. So with that, I will turn it over to you, Dr. Zhu.
Hi, thank you, Maria, for the kind introduction, and then welcome everybody. And also thank you for the North America for the opportunity to share our research. The title of my talk today is Dietary Sodium, Short-Chain Fatty Acid, and the Blood Pressure Control in Untreated Hypertensives. Sorry, I think I got, okay. I have nothing to disclose. I think we just learned very nicely from Dr. Lennon, gave a great overview of the gut microbiome in the human health. So as it's a new research area, and the emerging evidence shows the gut microbiome play an important role in the normal physiology and the disease process, such as hypertension. I think from the animal experiment, just as Dr. Lin just shared, so in the we can see there are differential fecal microbiome composition between salt-sensitive and salt-resistant rats. And also in spontaneously hypertensive rat, there's a decrease in gut microbiota diversity and richness. And also the study shows the high blood pressure appears to be transferable from hypertensive donor to germ-free mice through fecal microbiota transplantation. And in the humans, there are few human studies reported. You can see there are between the control pre-hypertensive hypertensive group, you can see the number of genes decrease. Also, the alpha diversity also reduced, and also compared to controls, the hypertensive group have distinct signature of gut microbacteria. There are many factors can affect gut microbiome, from the birth process and different infant feeding method, also to the life cycle stage. For example, when the adolescent goes through the puberty, and adult and until elderly, so also the life cycle have an effect. Another geographic location where you live also will affect your gut microbiome. And of course, the medicines will affect. Another lifestyle factors, for example, exercise, smoking, or psychological stress will also affect our gut microbiome. Diet is a very important factor affecting a gut microbiome. So sodium, as we all know, high sodium intake is a significant risk factor for hypertension and heart disease. And from the randomized controlled trial have demonstrated there is a causal relationship between the sodium intake and the hypertension. The high sodium intake increase your blood pressure. Lower your sodium intake, reduce your blood pressure. However, the underlying mechanisms are still not well understood. So this paper, Dr. Lennon just mentioned from the group, German group, was published in 2017 in Nature. So they investigate the effect of high salt diet on gut microbiome and the immune response. I think this is the first study explored the relationship between high salt diet and the gut microbiome. So I think Dr. Lennon just gave a detailed introduction. So I just briefly shows high salt diet alters the fecal microbiome. So between the non-intensive diet and the high salt diet, you see that several species got reduced. And also when you use the normal sodium diet, when after 2 weeks of high salt diet, you see the reduced the lactomarinus bacteria. When you stop the high salt diet, resume the normal sodium diet, the bacteria can recover. They also done that when you give the high sodium diet, they increase your blood pressure, the blood pressure in mice. While you, at the same time you supplement this bacteria, they can reduce ameliorate salt-sensitive hypertension and reduced the number of T helper 17 cells induced by high salt diet. And they also done, I think, the first human study in 12 male individuals. So they shown after 2 weeks of high salt diet, I think they gave them 4,000 microgram sodium on top of that their usual diet. So it's around 6,000 milligram sodium per day intake. After 2 weeks, those individuals, the blood pressure got increased. Also the T17 cell got increased. Also for the bacteria, the species got reduced. So the baseline, and then and the gray box is the after 2 week high salt diet. So those species either lost completely or reduced. And this is another study. It's a recent study actually. They look at it's a cross-sectional study of 135 healthy volunteers, and then based on their dietary is 2 instruments, food frequency questionnaires and the 3-day dietary recall. They determine their sodium intake based on this instrument. So they divided group between low sodium intake and the high sodium intake, and they see with the sodium intake increasing, there seems the patterns of gut bacteria profiling seems to be different. They also show the Shannon diversity seems decrease, although it's not significant, but there is significant in beta diversity. So short chain fatty acid are the metabolites produced by gut microbiota by fermenting the intake fiber, mainly in colon. So there are several types of short chain fatty acid. So acetate, butyrate, propionate are the three major types. Short chain fatty acid, they can serve as fuel for intestinal epithelial cells. Also, they can strengthen the gut barrier function. About 95% produced short chain fatty acid can be absorbed through intestinal epithelial transporters into the circulation. The absorption, the process can be regulated by sodium intake. The circulating short chain fatty acid can interact with the host protein receptors and then exert their effect on host physiology. So this figure was proposed by Dr. Plonzinick. So they demonstrate gut bacteria produce short chain fatty acid in the gut, mainly in the colon, through those receptors. Right now, I think there are at least 3 receptors has been identified can transport short chain fatty acid to the bloodstream, the circulation. So those short chain fatty acid in the circulation, they can bind to their G protein coupled receptors. There are at least 4 different types of G protein coupled receptors has been identified. So those receptors, they are expressed in colon, kidney, sympathetic nervous system, blood vessel, vasculature, and the immune cells. So through the interaction, that they can stimulate downstream cascade to regulate adiposity, blood pressure, immunity, also cell cycle, oncogenesis. The circulating the short chain fatty acid also can have direct inhibit histone deacylase activity to change chromatin structure and then lead to regulate gene expression. So they can also have effect on cell proliferation and apoptosis. There's one study, they show 4 weeks high salt diet reduced circulating level of short chain fatty acid, mainly butyrate. They also show high salt diet reduce the species of this bacteria. Also is short chain fatty acid producing bacteria. Also they changed the host immune system. So the knowledge gap, like Dr. Lennon mentioned, the studies in humans are limited. They're only limited to small sample size or very few in cross-sectional in nature. So very few studies in randomized controlled trials. Also, hypertensive patients has now been studied. So, so we hypothesize that dietary sodium reduction will increase circulating short chain fatty acid in untreated hypertensives. We also hypothesize that increases in short chain fatty acid induced by sodium reduction are associated with decreased blood pressure. So this was a collaboration project between our group and Professor He and Professor McGregor at Queen Mary University of London. They previously conducted a salt reduction study. It was a randomized, double-blind, placebo-controlled crossover study. So basically, at the baseline, the subject was assessed and their blood pressure was measured. 24-hour in 2 consecutive 24-hour urine were collected, and also blood sample were collected. At the visit, subjects were instructed to lower their dietary sodium intake. They gave some suggestions, the menus. Also, they were provided with free sodium bread. So after 2 weeks, the subject were randomized to 2 groups. One group will take slow sodium released tablet, 90 millimolar per day for 6 weeks. So on the sodium reduction, they aim to reduce their normal sodium intake to about 2,000 milligrams per day. So for the sodium tablet, they supplement 90 milligrams per day, equals to another 2,100 milligrams per day sodium for 6 weeks. And then they switch to placebo tablet for another 6 weeks. The second group will take placebo tablet for 6 weeks first, and then switch to slow release sodium tablet for 6 weeks. At the end of each 6 weeks, their blood pressure will be measured. Other cardiovascular phenotype will be collected, and then 2 consecutive 24-hour urine were also collected, and the blood sample were collected too. So their eligibility for their study is white and black and Asians aged 30 to 75 years old with sitting systolic blood pressure 140 to 170 millimeter mercury, or diastolic blood pressure from 90 to 105 millimeter mercury. Those hypertension were not on any antihypertensive medications. So they were original study was 71 white, 69 blacks, and 29 Asians completed the study. They already published in hypertension. They show that the modest sodium reduction for 6 weeks lead to reduced blood pressure. They also improved arterial stiffness. So we performed salt chain fatty acid profiling in stored serum samples, which will collect at the end of each slow sodium tablet period and placebo tablet period. The target salt chain fatty panel was performed by metabolome impaired serum samples from 145 untreated hypertension patients. These old samples available. So among those patients, 39% were white, 42% were blacks, 19% were Asian, and overall were 34% were females. So 8 salt chain fatty acids were quantified by liquid chromatography with tandem mass spectrometry. So the 8 mainly from group A and group B. So the statistical analysis was based on an intent to treat principle. And also we performed mixed linear regression to test the effect of sodium intake on the changes on short chain fatty acid, and we control for age, sex, race, and the BMI. So this table one shows among all 145 participants. So we have 49 females, 96 males. And after 6 weeks treatment in the slow sodium placebo, you can see their sodium, urine sodium was reduced. Their blood pressure was reduced, either office blood pressure and ambulatory blood pressure, or also their carotid femoral pathway velocity was also reduced to significantly reduced, and the pulse pressure also reduced in both men and women. And among the 8 short chain fatty acid we measured in the whole group, there are 4 of them was significantly increased. Actually, so almost everybody seems increased, but those 4 reached the statistical significance. So there are 2 methyl butyrate and butyrate, hexonate, and isobutyrate increased in the overall group. So the table shows exactly the levels, the numbers. These 4 significantly increased. So the other, you can see, also seems increased, but they did not reach a statistically significant. We also look at by sex. Let's see. Actually, to our surprise, we see all the significance was in the female group. So there were even 5 when we separate by sex. There were 5 short chain fatty acid was significantly increased by sodium reduction in the female group, including valerate, butyrate, hexonate, isobutyrate, isovalerate, and valerate. Beta represent a regression coefficient. We also look at to see whether the changes in circulating short chain fatty acid are associated with reduced blood pressure as well. And then we see, for example, isobutyrate, isovalerate, they see a significant association between the increased isovalerate by sodium reduction associated with decrease in blood pressure, in the ambulatory blood pressure, systolic, diastolic, and also there are a few markers of pulse pressure. So the PW and the pulse pressure, they are the markers for arterial stiffness, arterial compliance. So it suggests, so we observe a significant association between the increase in circulating short chain fatty acid and the blood pressure, and some markers of for arterial compliance. So the summary of our findings, so we found sodium reduction, actually modest reduction from their average, I think over 4,000 to 2,000 to 2,300 increased circulating short chain fatty acid in untreated hypertensives. Our study, I think, was the first to be able to establish the causal relationship by using the randomized controlled design. So we also find an increased circulating short chain fatty acid was associated with reduced BP. So this this is also a novel finding as well. We see direct relationship between the changes in circulating salt and fatty acid and blood pressure changes. And we also found sex differences. In the sodium reduction induced salt and fatty acid. So by looking at our finding, we compare our finding within the literature. So we found sodium reduction increased circulating salt and fatties in untreated hypertensives. In the literature, there is one study in rat. They show 4-week high salt diet decrease circulating salt and fatty acid, particularly butyrate. So we demonstrate sodium reduction is able to increase circulating salt and fatty acid. There is a very recent study published, even a relatively small cross-sectional studies, have 29 hypertensives and then 32 normotensives. They collect the fecal samples and then they measured salt and fatty acid in both fecal and circulation. So they shown in the circulation plasma, there are several salt and fatty acids levels was lower in hypertensive group compared to normotensive group, which is in agreement with our finding. We shown sodium reduction increase those salt and fatty acid. The three we have shown have been increased. For acetate, we shown the increase, but did not reach statistical significance. Interestingly, they measured since the salt and fatty acid in both fecal and the plasma at the same time. They show the opposite direction. In the fecal samples, in the hypertensive group, seem to have a higher level of butyrate, propionate, valerate, and acetate. So in the fecal sample, there are higher level of salt and fatty acid in hypertensive group, whereas in the circulation, they have lower level of salt and fatty acid in the hypertensive group, suggesting maybe hypertensive individuals or animals, they have impaired absorption of salt and fatty acid. And the recent study, I think that demonstrate they also measured the salt and fatty acid in both feces and the serum samples. In the juvenile and the adult rat, in the adult spontaneous hypertensive rat, there was an increase in feces butyrate level, whereas have a decreased circulating level of butyrate, which also I think in agreement with our findings. Data further shows there is impaired butyrate transport across proximal colon epithelium in the SHR. So which explains why they see the higher level in feces, but lower level in the circulation, because they have the transport process was impaired. So compared to normal rat, so HR have a lower basolateral butyrate level. They further see data on the gene expression and several transporters, but this one particularly is the transporter of butyrate. They saw the significant reduced gene expression of butyrate transporter SLC5A8. And the fluorescence staining also showed that at the number of cell membrane of the transporters, SLC5A8 was also reduced. I think this study illustrates in the hypertensive rat, the transporters numbers was reduced, gene expression was reduced, and the certain fatty acid absorption process was impaired and leads to reduced circulating certain fatty acid. Or another finding is we identify a sex differences in the sodium-induced certain fatty acid response. I haven't found any relative article in the literature to investigate the same question. But in the literature, there are a study shows there are sex differences in gut microbiota. The ratios of bacterial cell to human cells were different between males and females. And also this shows the gut microbiota is partly influenced by sex hormones in adolescence, maybe during the puberty period. The other study shows fiber has a sex-specific effect on gut microbiome, maybe due to their can affect estrogen level. This study shows there are sex difference between the microbiome between men and women. You see the difference, the relative abundance of different bacteria. And the different principal component coordinate analysis, also see they have distinct signatures. And another study I found that shows antibiotic exposure has a sex-dependent effect on gut microbiota and the metabolism of certain fatty acid and amino acid in mice. So this is a summary of our findings. So we have shown sodium reduction increased circulating certain fatty acid. So those increase also associated with reduced blood pressure and improved arterial compliance. This effect is especially significant in the female population. So there are sex differences. So this slide summary the mechanism how certain fatty acid regulate blood pressure. So there are many factors affect gut microbiota, especially diet. So sodium play a role in gut microbiota regulation and lead to changes of certain fatty acid. Those changed certain fatty acid can combine, interact with G protein coupled receptor at different tissues and organs and through different pathways, either through energy utilization, vasodilation, vasorestriction, and immunity, inflammation lead to blood pressure regulation. So future directions, like Dr. Lennon mentioned, large randomized controlled trials are needed to replicate, to confirm findings. Currently, there are only a few human studies and in the small scale, and very few in randomized controlled trials. So the large trials are needed. And presently for hypertension treatment in the future could be dietary intervention, sodium reduction, or increased fiber intake, or maybe probiotics could personally correct gut imbalance of microbiome. So also potential certain fatty acid intervention could potentially improve blood pressure as well. And sex-specific intervention should be considered when considering the treatment of hypertension. So our work was supported in part by American Heart Association. So our research has been supported mainly by National Institute of Health and American Heart Association. Like thank you for my postdoc and the colleagues at Medical College of Georgia, and my collaborators, Dr. He, Professor McGregor, and Dr. Wang at the Queen Mary's University of London. And thank you.